So this is a talk about our search for biomarkers of Alzheimer's disease and Down syndrome. And there are a few disclaimers in one of our pictures of Pittsburgh, but here's what I thought I would do. First, most of you probably don't know a lot about Alzheimer's, so we kind of figured I'd do a quick overview of what we know about Alzheimer's. And then we'll talk a little bit about Down syndrome for those who aren't well versed in Down syndrome, or at least Down syndrome adults. Most of us work with kids. And then in the remaining time, I'll share some findings and let you know kind of what we've discovered and kind of where we're headed. So let's talk about Alzheimer's first. So as you probably know, Alzheimer's is a progressive and eventually fatal neurodegenerative disorder. Symptoms include impact on cognition and memory, loss of activities of daily living. It affects a lot of people. 15% of individuals over the age of 65 in the US, which includes me, are diagnosed with Alzheimer's. I am not diagnosed with Alzheimer's, but I am over 65. And it's approaching 7 million Americans living with Alzheimer's. So this is a big group and a growing group. So how does this disease work? Well, hopefully all of us at this point are clinically normal, so we have no signs of Alzheimer's, and we hopefully have no disease or neuropathology in our brains that could lead to Alzheimer's. But the next step happens, and it's called the pre-symptomatic stage of Alzheimer's. So that's when our memory is still intact, we're still functioning well, but there are some early brain changes that can actually be seen. Finally, someone gets a diagnosis of mild cognitive impairment, or MCI, where there continue to be more brain changes, and we start to see the first symptoms. And then finally, one would get a diagnosis of Alzheimer's, mild, moderate, severe, where the brain pathology and neuropathology is quite classic and clear, and the symptoms are very apparent as well. So here's the take-home point. If you remember anything from the next 45 minutes or so, is that these neuropathological changes begin to occur 15 to 20 years before we see clinical symptoms. So what I tell all of our participants and all of their families is that if I'm going to get Alzheimer's in my middle to late 80s, the changes in my brain are happening right now as I'm talking to you. So what are these changes? Well, we know a lot about what's going on, and we think the first culprit is a protein called amyloid. And amyloid plaques deposit on the brain. They result from a couple of enzymes, first gamma secretase and then alpha secretase, that result in what are called amyloid plaques. Okay, and they start to deposit on the brain. That's the first step. And what follows is something that right now is referred to as the amyloid cascade hypothesis. And these are the things that happen for 15 to 20 years before we see symptoms. And so the first thing that happens is we can see changes in amyloid in cerebral spinal fluid. And then a few years later, we can detect it on a PET scan. And then a few years after that, we can see changes in a second protein that I haven't mentioned yet called tau. And tau leads to neurofibrillary tangles, which really eventually lead to neuronal death. And we can start to see tau in cerebral spinal fluid next, and then on PET scan. And then finally, we can actually see some structural changes in the brain on MRI. We can start to see changes in the size of the ventricles and hippocampal volume, which impacts memory. We can see it on something called an FDG PET scan, which measures glucose or sugar use or energy in the brain. And once that cascade happens, then we start to see some of the classic symptoms like changes in memory and functioning. So that's the amyloid cascade hypothesis. So I've already alluded to this already. And the question is, how do we see amyloid plaques in the living brain? Well, until about 20 years ago, the only way to see amyloid plaques in the brain was at autopsy, which obviously is too late. But in 2002, the first paper was published of a radioligand that could be used in a PET scan, and it was called Pittsburgh Compound B, or PIB, and it was invented by two Pittsburghers here at the University of Pittsburgh, Bill Klunk and Chet Mathis. And once they did that, they were able to use a structural MRI, a T1 MRI, and co-register it with a PET scan using Pittsburgh Compound B, and they could actually see amyloid in the brain, where it was and how much there was. And on these PET scans, amyloid is measured in SUV units, and orange shows a lot more amyloid, blue and purple is what we like. So here's a control of just an adult, not with Down syndrome, and you can see that there's no appreciable fibrillized amyloid in the brain. Here's someone with Alzheimer's, and you can see lots of red and orange in multiple regions of the brain. So this is what a PIB scan looks like. Since PIB was developed, there are other amyloid tracers that have been developed and are available commercially. And so probably a good half dozen or more of them are around, other than just PIB. And this, in addition, around 2014, another set of radio tracers to look at that other protein I mentioned, tau, also became available. And so now we can actually use radio tracers and look at amyloid deposition and the amount in the brain. And we can also use a tau radio tracer. The first one was AV1451. And you can see we can also look at the presence of tau. So we can now see this whole cascade before us. Okay, so by this time, you should be thinking to yourself and raising your hand and saying, well, Ben, if we know so much about the cascade hypothesis and what causes Alzheimer's, surely we've been able to make a lot of inroads towards preventing it and curing Alzheimer's. And the answer is no, we have not. So let's talk about that briefly. And hopefully I won't lose my voice because I have a week-long cold. I'll be popping my Luden's cough drops while we go along. So for the past couple of decades, there have been dozens, probably hundreds of trials of drugs that try to get rid of amyloid, that try to get rid of tau, that have been attempting to cure Alzheimer's. And until the last couple of years, they have essentially all failed. So let's talk about why they failed while I pop my Ludens here. Okay, well, in kind of an ironic twist, and this is simplifying things, but the main reason that most of these trials failed is because they enrolled people who had Alzheimer's, which is kind of what you would think you would want to do if you were testing out an Alzheimer's drug. But the problem is, if you've been following me, that by the time someone has Alzheimer's, they already have 15 to 20 years of the development of neuropathology. And by then, they're losing functioning. And you can eliminate amyloid, you can eliminate tau, but it doesn't seem to impact the course of the disease at that point. It's too late for us to intervene. So recently, in the last couple of years, 2 drugs have been approved by the FDA. You may have seen this in the paper or on television. And they seem to have slowed the progression of Alzheimer's. Why did they work? Because the population that they chose were people who had either mild cognitive impairment or just the early stages of dementia. And so there was a chance to make a change at that point. Where's the field going now? Well, the field is moving now to try to intervene before people are showing symptoms, the preclinical stage, because that's really where we can have the biggest impact. And that is kind of what we're going to be talking about. So just to bring you up to date, here's 3 of the drugs that are all monoclonal antibodies. That means they are all eliminating amyloid in the brain. Aducanumab was the first one, but I believe it was recently taken off the market by the drug company because Medicaid wasn't going to pay for it, so they're going to put their efforts elsewhere. So the only one currently approved is lecanemab, and another one, donanemab, is probably going to be approved later this year. And these were very controversial because the amount of improvement was not that significant. Essentially, it would give someone another 3 or 4 months of, if you looked at the change in curves, of clearer thinking before they started to have more significant symptoms. And that was a start, okay? So, but it wasn't a cure. So that's kind of where we are with these drugs. And there are now more trials looking at people, as we said, who early on who are not symptomatic but have a lot of risk factors for Alzheimer's. Okay, so let's talk about Down syndrome, which is why I'm kind of here today. Why is there some interest in Down syndrome when we think about Alzheimer's? All right, so to review, for those who don't remember, Down syndrome is otherwise known as trisomy 21. It's a triplication of the 21st chromosome, which impacts about 329 genes. Incidence in the United States is about 1 in 750 live births. So interestingly, the life expectancy of adults with Down syndrome has been increasing steadily over the last 70 years. Until now, it's over 60 years. But in 1950, and when I was born in the early 50s, I had a next door neighbor who actually had a preschooler with Down syndrome. And this was very unusual in 1950 because most pediatricians would tell a parent, if they gave birth to a child with Down syndrome, that they should institutionalize them. But even though he was living at home, his life expectancy was still only in the teens. But it has gone up steadily. And it's gone up for a number of reasons. One, the institutionalization, early intervention, good education throughout 0 to 21 years of age, addressing medical issues more aggressively, like sleep apnea, hypothyroidism, celiac, overweight, those types of things. And then a major one that really had an impact was around the 1970s, changing kind of the standards of how to treat a early cardiac problem. Many children with Down syndrome, probably 40%, were born with a heart defect. It was a valve defect that in any other child would have been easily corrected very shortly after birth. But at that point, people said, well, why would we do that for infants with Down syndrome? They're not going to have a quality of life anyway. Well, that thinking changed, and that also has resulted in this increase in life expectancy. Okay, so as people with Down syndrome started living longer, we noticed that they seem to be at high risk for early onset of Alzheimer's. Early onset means Alzheimer's before the age of 65. And in fact, virtually every adult with Down syndrome will have the neuropathological changes that are consistent with Alzheimer's by their early 40s. And most will develop dementia by their late 60s. So here's a cumulative curve of the rate of dementia, the cumulative incidence of dementia in Down syndrome. My colleague Nicole Shoup from Columbia published this. So you can see that until about the middle 40s, no one with Down syndrome has dementia. So if some of us are seeing an individual who's 20, 25 years old and suddenly has some significant changes in functioning and they have Down syndrome, it is not dementia. It's something either we call catatonia or Down syndrome regression or some other medical issue, psychiatric or a stroke or a tumor, but it is not dementia. But then you can see by the middle 40s, we start to see a slight uptick in the rate of dementia diagnoses, and it goes up pretty quickly after that. The mean age for a diagnosis of dementia for adults with Down syndrome is somewhere between about 53 to 57 years of age, depending on the cohort. So that's the average, the mean age. So why are adults with Down syndrome, for the most part, all getting dementia? And the answer is fairly simple. Remember, we talked about this APP precursor protein, which makes amyloid. It happens to fall on the 21st chromosome. So that means that someone with Down syndrome is making 50% more amyloid than someone in the general population from probably in utero. And so by the age of 40, it's pretty much overwhelming the system. And we just can't, individuals with Down syndrome just can't get rid of the amyloid quick enough, and it starts to build up. And so we see early signs of dementia. Okay, well, let's talk about what we've been doing for the last really more than 10 years, probably about 15 years. Let me tell you a little bit about how we got started in this, because it was kind of serendipitous, which is the case in most of my life, I think. But we were doing, I was doing a study, a drug study with adults with Down syndrome. In fact, I was doing it over at Cat Adult in around 2004, 2005. We were studying a drug called Aricept. Now, some of you may have heard of Aricept. It's actually a drug to treat Alzheimer's. It doesn't prevent Alzheimer's, but it adds some acetylcholine in the system so that thinking, memory skills are maintained for a slightly longer period of time. And we weren't studying adults with Down syndrome who had dementia, but the thought was maybe Aricept would improve cognition in young adults who didn't have dementia. Well, it didn't, but it just turned out that a couple people that we've already talked about had recently developed this radio tracer called PIV. And that group approached me and said, you know, we have this radio tracer, and people with Down syndrome all should have a lot of amyloid in their brain, but no one's ever looked at it. Do you think we could get them to cooperate with an MRI and a PET scan? He said, of course, why wouldn't an adult with Down syndrome cooperate for an MRI and a PET scan? They would go through lots of procedures. It would be easy. And so what we did is we were able to get 8 folks, 7 who were willing to cooperate. They all were doing fine. They were all cognitively stable. They were fairly young, 20 to 44 years of age. And they all sat in for a structural MRI and then an amyloid PET scan and then a cognitive assessment. And this is what we found. These are our 5 youngest folks. So you can see they're 20, 22, 34, and 35 years of age. And you guys are all now experts at reading amyloid PET scans, right? So you can see this is what we hope all our brains look like. They're all, don't have any fibrillized amyloid. Okay, but what about our older participants? And we don't mean like a lot older. I mean 3 years older, 38 and 44. And you can see here that we're starting to see in our 38-year-old lots of yellow and some orange, indicating we're seeing amyloid deposition. And here's our 44-year-old who has amyloid in multiple regions of the brain. In fact, one of the Alzheimer's researchers looked at this and just assumed this was someone who had Alzheimer's, but in fact, he was functioning perfectly fine. Let me also point out 2 regions here. You can see we have some developing kind of red here and really obvious here, which are where most of the amyloid seems to be going. That's in the striatum. This is an area that seems to be the first area that is affected in Down syndrome. And it was interesting at the time because there are a couple other genetic disorders that lead to Alzheimer's, as does Down syndrome. And we see the same pattern in those disorders. They're called autosomal dominant genetic disorders. And they also have the same pattern. This is not the pattern you would see in an 85-year-old who has Alzheimer's. So we published the paper. It was one of the first 2 papers in looking at amyloid PET scans in Down syndrome. So that we used that data, and we got our first funding in 2009 with what was called an R01 from National Institute of Aging. And then in 2015, we got our first grant to do ABCDS, or the Alzheimer's Biomarker Consortium. And then in 2020, we got our follow-up grant, which is now a U19. So ABCDS now has 9 sites across the US and the UK. And I'll just mention the clinical sites: University of Cambridge, my favorite site because I like to go visit; Mass General Hospital; Columbia University in collaboration with Institute for Basic Research; us at the University of Pittsburgh, we're what's called the Coordinating Center; University of Kentucky; University of Wisconsin in Madison, Washington University in St. Louis, University of Kansas Medical Center, and University of California in Irvine. And then you can see in blue, we have a lot of other associated sites that help out with genetics, analysis of data, storage of blood samples, brain samples, et cetera. So it's a big project, as you can see. So here's how you get in the study currently. Obviously, you have to have Down syndrome. There are 3 different types of Down syndrome. There's full trisomy, there's partial trisomy, or where part of the 21st chromosome is replicated, not the entire. And there's mosaic. And we take everyone. Some studies only take full trisomy. We're interested in everyone. You have to be 25 years of age. Mental age has to be greater than 3 years because we find that's what you need to be able to do most of our cognitive tasks. Right now, we're taking only healthy individuals, those who do not have Alzheimer's yet. But if someone has been followed in our study and has Alzheimer's, we will continue to follow them. And we need a caregiver serving as a reporter. We also have a small group of sibling controls. And we have folks come in every 16 months. So we are now up to a lot of folks in our 5th cycle. First person came back for their 6th cycle. So this is from 2015, so the last 10 years. The visits are rather involved, 2 or 3 days here in Pittsburgh if someone comes here. Just a sense of what we're gathering. We're gathering blood to do genetics, such as a GWAS or whole genome sequencing. We karyotype, meaning we confirm that they have Down syndrome, and we look for some other genetic biomarkers, something called APOE, which in certain alleles makes one more at risk for Alzheimer's. We have a neuropsychological cognitive assessment. The caregivers are interviewed and fill out a lot of questionnaires about functioning. We have a physical and neuro exam. We take blood for proteomics, measuring proteins in the blood, and metabolomics, looking at various metabolites in the blood that could be involved with inflammation or other types of biomarkers we'll talk about. We do an MRI scan. We try to get up to 6 different sequences in if someone will cooperate that long. We do an amyloid PET scan, which obviously makes sense. We do a tau PET scan. We do an FDG PET scan, which looks at, as I mentioned before, glucose use, energy use in the brain. And for those who are willing, we'll do a lumbar puncture to get cerebral spinal fluid. So it's quite a protocol. So how we've been doing? Well, this is our enrollment as of January over the last 9 years, 8 years. We've had, and again, this is across all 9 of our sites. We haven't seen this many in Pittsburgh. Cycle 1, our first visit, we've had 536 folks come in for a first visit. 360 have returned for at least 1 visit. 290 have come back for 2 visits. 163 for 4 visits. 26 have had 5 visits. And I didn't have space, but we had our first 6th cycle visit earlier in the year. So 1,375 visits. So what are our goals? What are we interested in looking at? Well, obviously, we're looking at the course of biomarkers and how they change with age. And the goal is to identify biomarkers that signal future onset of mild cognitive impairment or dementia. We're interested in looking at possible protective factors because, again, as you saw, people with Down syndrome can get Alzheimer's as early as the late 40s to the 60s. So that's a wide age span. There's got to be a reason why some people can hold off and maintain good functioning into their 60s and others start to lose functioning in their late 40s. So there may be some protective factors or other risk factors that we need to look at. We're looking for neuropsychological or cognitive measures that can help us detect the earliest signs of dementia. And all of this together is to lead to possible targets for future treatment trials. And in fact, Down syndrome trials, prevention trials are starting now. We are trying to, working to get into our first one and hope to join a couple of others before the year is out. Okay, so before we start to share some data, because you're kind of thinking, oh yeah, when do you actually have something to show here? Let me just give you, because you may not have met anybody with Down syndrome who has dementia. What does it look like? And I just thought I would share this one slide to kind of give you a sense of what the impact is. So this is a gentleman that I met. He was 53 years old, and this is a copying task. And at 53, he was cognitively stable. He wasn't able to copy enough to actually earn a point, but this was a pretty good attempt. And when he came back 3 years later, he had mild cognitive impairment, and you can see that his copying skills had started to deteriorate. And by age 58, when we saw him 2 years later, he clearly had dementia, and his attempt to copy was just quite poor. So that's the impact of dementia. Okay, so we have some time. Let me go through a few slides to kind of show you some of the things that we learned. Maybe leave a little time for questions. So one of the first things I was interested in, I think we all were interested in the study, was when do adults with Down syndrome actually start depositing amyloid, which is the first step in this cascade. The neuropathologist who had looked at brains of people who had passed away were telling us that sometime in the early 40s, people with Down syndrome had the neuropathology associated with Alzheimer's. So what we do with the amyloid PET scan is we have a cutoff to decide whether someone is amyloid positive, meaning they have a significant amount of amyloid in their brain, or amyloid negative, they're below threshold. And so this is by age group, about the percent of people who are amyloid positive. And you can see that under the age of 30, nobody is going to be amyloid positive. This is already 255 individuals. It's just, you're not going to see it. And again, I go back to the issue of you see somebody who's 25 years old, they've suddenly been losing functioning. This is not dementia, at least Alzheimer's type dementia. Even in the young 30s, middle and late 30s, the chances of seeing someone who's amyloid positive is pretty rare. But look what happens once we get into our early 40s. Over 50% of people in their early 40s become amyloid positive, over 80% in the middle to late 40s, approaching 90% in the 50s, and by the middle 50s, 100% of the people we've seen are amyloid positive. So this, I just wanted to also illustrate, hopefully my pointer's working, that when we think about drug trials in Down syndrome, it's going to be a challenge, as my team and I think about this, in terms of it's a very small age group that we can enroll. So one of the trials that we hope to join, and that will happen, would be a trial with one of these new drugs, an amyloid, a drug, a monoclonal antibody that eliminates amyloid in the brain. Well, to get in a trial like that, you would have to be positive. You would have to have amyloid in the brain. It wouldn't make sense to give somebody a monoclonal antibody if there wasn't any amyloid to get rid of. So that means that you really can't enroll anybody in their 30s. There's hardly anybody there. And only half the people in their early 40s are going to be amyloid positive. And so your best group are the 45 to 50, maybe up to 55-year-olds. But then once you get into your 50s, a lot of people, over half, are going to have dementia already. And those are not folks you're going to enter into the trial, because we already know that these drugs are not going to help those folks. So we have this very narrow age range that we're going to be able to recruit from. So it's going to be a challenge. It's going to take a lot of different universities across the world, really, to pull this off. Okay, I've still got my voice. So another question we were asking and interested in, this was a hypothesis I kind of wrote out in one of the early grants, and we finally got a chance to test it out. And within the general Alzheimer's population, there's a concept of something called cognitive reserve. And, excuse me, that suggests that, and has been shown, that people who are college educated tend to be able to maintain cognitive functioning a bit longer than people with high school educations who might have the same level of neuropathology. So the thought is we just have other ways to work around problems with memory and cognitive functioning, and we can hold off for a number of months more, maybe a few more years. So the thought was maybe we'd see the same effect in Down syndrome. Maybe those who had mild intellectual disability might develop Alzheimer's a little later than those who had moderate intellectual disability or severe intellectual disability. So we finally were able to look at it with about 360 plus individuals. And here's how we broke them down. This was just published a couple of years ago by my colleague, Segan Hartley of Wisconsin. And we obviously looked at a number of measures that you're now familiar with, amyloid PET and tau PET. And our cognitive measures, one was called the acute recall test. It's a test of episodic memory. I'm going to show you that in a second because we use it a lot. Block design, those of you who are psychologists, remember these are blocks, and we create a design, and the individual, the child or adult, has to copy the design. And then there's something called the Down syndrome mental status exam. This is about a 20-minute test that it's just a general mental status exam. It asks about memory and motor functioning and visual spatial skills, and you get a score. So those are the measures we were looking at. Let me tell you a little bit about cued recall and how this works. So cued recall involves teaching an adult with Down syndrome to remember 12 pictures, and we present 4 at a time. So on 3 different plates, 4 pictures. And after they learn the pictures, we turn all the plates over and say, tell us how many pictures you can remember. And that's called free recall. If they can't remember a picture, we give them what's called a cue that we've also taught them. So let's say in this picture, they couldn't remember the helicopter. So the cue would be, it's something that flies. And they say, oh, that's a helicopter. That's a cued recall, and that counts as well. So the total score could be up to 36 points, free recall and cued recall. And then we also look at intrusions, and those are essentially incorrect recalled pictures. So someone might not remember grapes, but I would give them the cue and say it was a fruit. And if they say, oh, an apple, that would be an intrusion. Okay. Okay, so let's look at some data. So we didn't expect to see anything here, and we didn't, which was good. So this is the amount of amyloid using a scale called centiloids, and our blue line are those with mild ID, red have moderate intellectual disability, and green are severe. And here's our age, and here's their amount of amyloid. And obviously, nobody's going to have, just as you figured out, no one under the age of 40, for the most part, is going to have much amyloid, okay? And then once they reach their 40s, everybody goes up. But the important thing is regardless of IQ level, everybody's going up at the same rate. And we wouldn't expect that people with different IQs would accumulate amyloid at a different rate, but we just wanted to be sure that was accurate. And here's our tau protein. Again, our measure of tau, and we see the same thing. As people get into their 40s, the amount of tau goes up, but these lines are parallel, pretty much together. So no difference on IQ. But what about on our cognitive measures? So here's our cued recall score. That's our episodic memory score with a maximum score of 36. So obviously, the green line, which are individuals with severe intellectual disability, are not going to score as well, regardless, as those who have mild or moderate. But you can see that as they get into their 40s, everybody's memory starts to decrease, but the lines are all parallel. It's not like those with mild intellectual disability were able to maintain skills and they didn't start to lose until later. They all look like they're the same. When we look at block design, we see the same thing. People are starting to lose block design. Their scores go down, but they're all parallel. So they're all going down, starting to go down at the same time. And Down syndrome mental status, the same exact thing. So what we found out was there's not a similar concept of cognitive reserve in Down syndrome. Okay, doing good. Okay, so let's go back and look at some amyloid PET scans. So as we started having people come back, we also saw some interesting things. And here's each of these is an individual person. So this person came back 3 times, and here you can see his amyloid load first time was still high because this is our cutoff for being amyloid positive. So the folks in red started out amyloid positive, and obviously no one lost amyloid when they came back. They continued. And so you can see it goes up the next time and continues to go up. And everybody's rate of change is pretty similar. It's a fairly steep climb. But we also saw folks who started out amyloid negative in blue, and they came back and they remained amyloid negative. They remained below the line. And you can see that it's, as they start to accumulate around the younger folks, age 30, around 45. And then we have folks who were converters. They were folks in black who started out with at least their first visit or their first couple of visits being amyloid negative, but then at some point in one of the visits, they crossed the line and became converters. And if you look at their rate of change, it's very similar to the rate of change of those who started out amyloid positive. They just started out lower. We caught them earlier. Okay, but you can also see how age is affected and how this is so widely distributed. Okay, so this is kind of messy, and you can see this is similar to the previous figure that I showed you. A couple of grad students or actually postdocs, I guess I should say they're postdocs now at Wisconsin, came up with something called sampled iterative local approximation trajectory modeling. I hope anyone will ask me to explain that. But essentially, what this allowed us to do is to try to bring this data closer together. And so what they did was they created this 0 point here. The 0 point is where someone becomes amyloid positive. And if we know when they became amyloid positive, it's fairly simple. You kind of just move the lines over. But what about the folks who haven't become amyloid positive yet? So this SILA modeling allows us to project when that will be. And you can see we come up with a very clear line of what amyloid is going to look like after someone becomes amyloid positive, okay? So people become positive anywhere across like a 15-year age span, but once they become positive, this is in general what the acceleration is going to look like. And what we also found in this paper was that the change was about 7 units a year. And that's faster than what we find in what's called late onset Alzheimer's, or people who get Alzheimer's in their 70s and 80s. They're accumulating at about 4 to 5 centiloid units per year. Folks with Down syndrome seem to be accumulating considerably faster. We do intolerance. Okay, so this is another study the team in Wisconsin looked at, and that was trying to figure out, remember this cascade, we have amyloid and then we have the protein tau. So the question was, when does tau, once amyloid becomes positive, how long does it take before tau in this cascade becomes positive? Is it years, months, days? Well, what they found out was that they measured tau in 6 different Brock regions, or Brock stages, and each Brock stage covers different regions of the brain. The one that seems to be affected first is the Brock 1, and that's the entorhinal cortex and the hippocampus. Hippocampus is involved with a lot with memory, so you would imagine that might be the first area. So what they did is they looked at folks, and we often do the tau and the PET scan at the same time, same day or the same visit. And so they looked at people whose amyloid PETs became positive. What did their tau PET look like? And you can see that's 0 years. That's this first group. And so when you're first diagnosed being amyloid positive, there's not a lot of change in tau. But within 2.5 years, the next time you come in, essentially, or the 2 more times when you come in, because we see folks at 16-month intervals, but within 2.5 years, when they come in, we see a real increase in Brock region 1 and starting to see increases in tau in these other regions. And within 5 years, we start to see the other regions, the Brock regions, also increase in tau. And so what we're seeing is that the point is, when you're going, well, why are we showing this? Is because this is much faster than in the general Alzheimer's population. You wouldn't expect within 2.5 years to see an increase in tau once you become amyloid positive. So this just continues to give us some data to show that this seems to be an earlier process in Down syndrome and a faster cascade in Down syndrome once it starts. Okay. Let me talk a little bit about some other biomarkers. We've mostly been talking about PET scans, but really where the field is headed are what are called plasma biomarkers, because these are biomarkers that we can see in the blood, which makes it much easier. It means any of us can have our doctors follow us, they can do a blood test, and they can measure these biomarkers without having to do an expensive brain scan or having to draw cerebrospinal fluid. So here are some of the plasma biomarkers that the field is really interested in at the moment. One is a tau one called p-tau217. Tau is phosphorylated in a lot of different areas, but this particular one, 217, seems to be the best predictor of cognition and change. There's another one called glial fibrillary acidic protein, GFAP, which is a measure of kind of neurodegeneration. We have some measures of amyloid, and there's one called neurofilament light chain, or NFL. I like that term, NFL, which seems to be a measure of inflammation, but it's not doesn't seem to be specific for Alzheimer's. You get inflammation with lots of diseases. So here's just a quick look at some data from some colleagues in Sweden who we shared data with, and they looked at this plasma p-tau217, which is a measure of tau in the blood system. And we looked at the sibling controls, all of who were amyloid negative, okay? Because they're the same age as the adults with Down syndrome. Why would they have a lot of amyloid? They're 40, 50 years old, right? And we compared them to the adults with Down syndrome, who also were amyloid negative. And you can see there really isn't, well, there is actually, a slight difference, but there really isn't a significant difference in terms of their tau. And then here's the folks with Down syndrome who are amyloid positive, and you can see their p-tau217 is significantly higher. So it kind of shows us that we can measure, we can get a sense of what the amyloid level is by looking at p-tau217. And the same thing with GFAP, which is kind of a measure of neurodegeneration. Again, our amyloid negative groups, the siblings and those with Down syndrome, generally still the same, but once you're amyloid positive, measures of plasma-based neurodegeneration also go up. Okay, I'm going to share one more piece of data because I'm kind of excited about this. This was just shared with us in San Diego at the end of January at our annual meeting by one of the postdocs two of the postdocs in Wisconsin. And they looked at the relationship between what's called amyloid chronicity. That's that 0 point when someone becomes amyloid positive, and its relationship to 4 cognitive measures. We looked at it cross-sectionally and also longitudinally. So let me explain what this is. So here's our measures. Here's our cued recall total score. You know this by now, it's 36 points. This is our intrusions for cued recall. This is the Down syndrome mental status exam. And this is the score on a parent completed questionnaire about dementia symptoms called the NTG. Okay, so our question was, here's our 0 point that we talked about when people become amyloid positive. The question was, at what point do we see changes in this curve when they become amyloid positive? And so what they found out was that once out on the cued recall test, okay, of episodic memory, within about just under 3 years, once someone became amyloid positive, within about 3 years later, we would start to see changes in memory, okay? And we also saw a similar change in intrusions or incorrect responses about 3 years later. We didn't see any statistically significant change in the curve with the mental status exam, probably because it measures so many different areas of cognition that any changes in episodic memory, which are included, just got washed out by things that hadn't changed yet. And interestingly, the parent completed or caregiver completed questionnaire 3.5 years after someone became amyloid positive, we started to see a significant change in the curve showing decrements in functioning. So first cognition, then change in parent report. We tried the same thing with longitudinal data because cross-sectional data, doesn't follow someone. And we have this great study where we can actually look at actual people and how they changed. So we wanted to see if it held up. And so we have our 0 point here, and then this is change. So 0 means that there wasn't any change when people came back. And again, it held up. It was about the same. It was about 3.4 years after someone became amyloid positive, we started to see change in memory. Didn't hold up with intrusions, but it did hold up with the parent report. But the interesting thing to me is the parent report was 5.5 years. So once someone became amyloid positive, it took over 5 years before the caregiver started to see clinical changes, which makes sense, right? Because we know that these things are going to, changes in neuropathology are going to happen before we see symptoms. But on cognition, we have more sensitive measures. We could start to see change at 3.5 years, 2 years before caregivers started to see changes. So with that, I'm going to stop because we have 5 minutes for questions. How about it? Or what do you think? Should I keep going, Carla, or leave time for questions?
I don't know if people want to chime in and say if they have questions, but I'm enjoying hearing what you have to say.
All right, well, I'll keep going. People
Yeah.
know where to find me. But,
Yes.
all right, let me tell you a little bit about some other things that are happening in Alzheimer's and what we're doing in Down syndrome. There is something called a staging model. It's called ATN, and it follows this cascade that we've been talking about. So the A stands for amyloid, which we can measure by PET or cerebrospinal fluid or with blood. T stands for tau, measured the same way. And the N stands for neurodegeneration. So the typical way to do it is hippocampal volume or FDG PET, change in glucose use, or maybe GFAP, which is a measure we can get on plasma. So they're staging thinking that depending on whether someone is positive in A, positive in T, positive in N, tells us kind of where they are in kind of the dementia journey, kind of like we stage cancer. And now the field is even thinking of expanding this to include a few other biomarkers, such as an I for neuroinflammation or a C for cardiovascular disease. So next year, maybe it'll be AITNC, but at the moment it's ATN. So we were interested in looking at the ATN as a staging model in Down syndrome. And we obviously had the A already for amyloid. So someone was either amyloid positive or negative, depending on cutoff. We had our tau measure based on our tau PET scan. And we also decided to we had a structural MRI, so we could come up with hippocampal volume measure. The problem is that you can't use a hippocampal volume cutoff with the general population because people with Down syndrome's brains generally are smaller than those in the general population. So we had to come up with our own cutoff. And what we did is we took the folks who were cognitively stable, found their mean, and adjusted it for what's called intracranial volume, and decided that anyone whose volume was more than a standard deviation lower than the stable group, we would call them N positive. So you can argue and say that doesn't make sense, but we did it. So we ended up with 4 different groups, if you can think about this. So we have the group that are amyloid negative, tau negative, and neurodegeneration negative, if you remember. And if you're amyloid negative, you're probably tau negative because you have this cascade. It's unusual to be amyloid negative but tau positive. It happens occasionally, but that would be unusual. Then we have folks who are amyloid positive but negative for T and N. And then we have folks who are amyloid positive, tau positive, but not neurodegeneration. And then folks who are positive for everything. And this is our cued recall total. You guys know cued recall by now. 36 points is the max. And you can see the mean for those who had no positive on all three was about, was about 36. And even if you became amyloid positive, that was not enough to significantly impact your memory. But once you are amyloid and tau positive, now we start to see a drop in cued recall, and it continues to drop when all three areas are positive. And if you look at intrusions, you see the same pattern. So this ATN staging profile seems to work in Down syndrome as well. I will end. Oh my God. All right. Well, I'll end with what we have a couple papers out where we compared Down syndrome with some other groups. And I mentioned this group earlier called autosomal dominant. This is another genetic disorder that also results in early onset Alzheimer's. And we compared amyloid PET scores with the Down syndrome group and this autosomal dominant group. And again, you can see those who are amyloid who are asymptomatic look pretty much the same. Down syndrome, same amount of amyloid PET as those with this other group, and those who are symptomatic have higher rates of amyloid. So I'm going to end with that. I got to thank, let's see, well, I'll tell you what we're doing next. We're going to continue. We're writing our continuation grant later this year. And for me, the big news is we're involved in prevention trials, and we hope to follow folks for years after they've been in a trial to see what happens, because a 12-month trial of a drug to get rid of amyloid, that may be great, but does the amyloid come back? Are you good for another 50,000 miles? What happens? So we really need to follow folks. So I'm going to thank all of our participants and families, and those of you who do research. This is our sites, our core leads. There are 94 co-investigators. It's a big group.